So hi everyone, I think we should start our session since it's 2:20. I'm here with my colleague Trevor Rashgosh, who is the Business Development Manager and Strategic Manager at Perceive AI. Myself, Christian Osro, I'm the CEO and co-founder of Perceive AI. So thank you very much, all of you, to attend this roundtable. The topic of the roundtable is improving treatment efficacy and de-risking clinical trial with precision medicine. So I'm going to try to cover some of the basis and a little bit what are our approach regarding that topic. And after that, we will try to have an open discussion where you will be able to raise your hand, and we will give you the authorization to basically ask your questions or give your inputs on various questions. Thank you. So patients are very diverse and disease are very complex. And that leads, unfortunately, to a lot of heterogeneity in the disease trajectory and in the treatment response of those patients. And that heterogeneity will have a lot of causes or problems downstream. So you will have patient variability that will lead to the researcher applying or evaluating a drug on the wrong patient. So on top of all the inclusion-exclusion criteria that you will find in a normal clinical trial, unfortunately, you will end up with a population that is still very diverse and heterogeneous, and that will lead to a poor patient selection basically in the trial. That will cause many downstream effects. So you will have high failure rates of those trials due to that, a massive cost of those trials, especially in late phase trials. Long trial duration, normal trials, especially the late phase ones, can go up to 5 to 7 years. And low treatment efficacy and poor patient outcome once those drugs are in the clinic or are approved and therefore used on these very heterogeneous population. So the basic concept of precision medicine is to treat the right, is to use the right treatment for the right patient at the right time. And this is a fundamental concept that is not new, but it took a lot of time to start to see emerging solutions that do apply that and makes it a reality either in the clinical trial side or on the clinical side. So prognostic and predictive enrichment strategy are fundamental tools for enhancing clinical trials and embracing precision medicine. There are two main layers for predicting health outcome. So the first one is prognostic enrichment. Basically, this is selecting patient with a greater likelihood of having a disease-related event. So basically, you want to identify individuals that will have an event or that will progress in their disease during the duration of the trial. Any subjects that will not display those properties will unfortunately not be suitable to demonstrate that your drug that you're trying to apply to those patients will be effective. The second layer is predictive enrichment. So the definition of predictive enrichment is to select patients who are more likely to respond to an intervention drug. So in this case, you want to further refine your selection. Once you have some data on the people that have been treated with the drug, you want to try to identify what are the characteristics that define those individuals that will have a positive response. And on the reverse, you can also identify individuals that will have an adverse event or that will just not respond to that specific drug. And therefore, you have a framework to really identify more suitable population for treatment. So it has a lot of impacts, basically, that will come with these types of enrichment strategies. So you can basically prevent endpoint failure in clinical trials, lowering the cost of bringing in new drugs to the market by reducing either the duration of the trial or the sample size needed to demonstrate that your drug is effective. Reducing the time spent on clinical trial recruitment by having a more automated way to identify those individuals that will have the right profile. And finally, improving treatment efficacy by having a more a subgroup of individuals that have been targeted and well defined to be responding to that specific treatment. So health outcome prediction requires a more complete understanding of the patient, a broader view of exactly what characterized that patient. And that comes with the use of multiple modality to characterize those individuals. So it can be imaging, genetic, clinical workup, blood tests to really have a full view of the individual and therefore being able to predict its future evolution. Obviously, multimodal data is extremely complex. Some of it are structured data, others are unstructured data. And this is very difficult for a human to really make sense and wrap his head around all that data. Although that said, it's a very well-suited work for machine learning or deep learning solutions. And it's really made to actually ingest these types of data and make sense of that plurality of features. So here I'm going to show some of the, one of the applications that we do at Perceive. It's in Alzheimer's disease. So Alzheimer's disease has a very long history of failure in clinical trials. In the past 20 years, we had 99.6% of clinical trials that did fail, unfortunately, for various reasons, among which the variability of the patient recruited. You still have, with the inclusion-exclusion criteria, a large proportion of individuals that do not decline cognitively during the duration of the trial, which is very important point. Individuals need to decline cognitively during the duration of the trial to demonstrate that your drug is actually slowing down or stopping that decline. So what we've done is to devise an enrichment tool for cognitive decline. So we target individuals at baseline and we try to identify the one that will be at higher risk and lower risk of cognitive decline. So from that very heterogeneous population, using our Foresight platform, that combines multiple modalities, we are able to identify a subgroup of individuals that will be or remain stable for a few years, and therefore we can enrich the population with individuals that will actually have a more drastic effect and more drastic cognitive decline. So that has multiple impacts, among which we've been able to demonstrate that we can reduce the duration of the trial. So see significant changes instead of 3 years, but 2 years in the trial, and also reduce the sample size of that trial by 30%. So you need less individuals to demonstrate the same effect. So this is just a basic overview, and I would like to open it to questions and discussion on enrichment strategy, and maybe give the mic to Trevor so that he can relay either your questions, or if you want to ask your question orally, you can raise your hand and we will give you the rights to speak. Thank you very much.
Great, thank you so much, Christian. So I see many people joined us part of the way through. So just a quick recap, we are Perceive AI. We are an AI-driven precision medicine company, accelerated clinical research. And so the goal here is to have an interactive discussion. And so just raise your hand or type your question in the Q&A. And we're more than happy to have a back and forth. So just to kick things off, Christian, one question that I had was, how can a better understanding of patient heterogeneity help researchers in medical fields?
So basically, one of the things that is, so I've presented more application in the clinical trial setup, but obviously those tools, once you have a better characterization of individuals that will be at higher risk or that will respond better to treatment, can definitely be a super useful tool in the clinic for physician, either to better manage their patient, the visits, like if you know that an individuals will decline cognitively in the near future compared to somebody that will remain stable for, let's say, the next 2 years, it's a very useful tool to manage when you will set up your next visit, or to provide adequate care to that individuals if he needs more support or specific facilities that could be extremely useful. And also just to take a better decision on which drug are potentially the most effective for that specific patient.
Very interesting. Thank you so much. So here's another question. So building from
We
what
have a few
Dr. Oh, sorry, go ahead.
questions already. Let me just check. Try to do both at the same time.
Sure.
Sorry about that.
All right. So we have a question from Samuel. Wouldn't these tools introduce selection bias into the treated population, therefore only work for narrow indications trial?
Can you repeat the question?
Sure. So Samuel says, wouldn't these tools introduce selection bias into the treated population, therefore only work for narrow indications trial?
So obviously, like you narrow down a little bit the population that will be treated. That said, for indication like very complex indications like Alzheimer's disease or cardiovascular, where you have like a very large spectrum of individual that will have like very different behavior. For the moment, it's better to have a more clearer, well-defined group of population that do respond well to the treatment, and then expand from that to a larger population. For the moment, what is clear is that basically trying to do the one-size-fits-all doesn't work in these types of complex disease. So we need to be a little bit more clever and pinpoint subgroups of individuals that do have the correct properties and the right properties for treatment.
And just to add to that also, having a better understanding of the patients treated is in general a very good thing and allows for more personalized treatment plans going down the road. So thank you very much for your question, Samuel. Here's a question from Jackie, very timely and very interesting question. How does our technology take into account racial, genetic, and gender differences between patients? Clinical trials have historically focused primarily on white males. How can you impact that?
It's actually a very good question. I don't have like a perfect answer for that. Like these types of features are included in our model to try to modelize that. The fact is, and this is an issue that will hopefully be reduced in the future as we increase the spectrum of individuals that are either in the clinic or of the data set that we are using for training of those algorithms. For the moment, we have to deal with the data that we have. So either the public initiatives or the clinical trials to train those algorithms. That said, there is various strategy that you can use more in an unsupervised way to try to compensate for these types of bias. But it's still like, it's important that those individuals or groups do participate in either in the clinical trials or in public initiatives to really have a better idea of how these groups do behave in reality.
Great point, Christian. Thank you so much. Here's a question from Sandeep. Have you been approached with having your foresight solution in the conduct of a clinical trial with a master protocol, which is a newer emerging model of clinical study, yet by a sponsor, CRO, or national collaboration, example in oncology research?
So we don't do oncology currently. We've focused on more like broad disease like Alzheimer's, and now most recently in cardiovascular. So I'm not as familiar with these type of protocols. Trevor, are you a bit more familiar? Maybe this is an answer that you can.
So there's increasingly, especially we're looking at the importance of decentralized trials, especially since COVID has halted enrollment for a lot. And so as things are very unevenly reopening in different parts of the world, it's very important to have a collaborative workflow. And so a benefit to our model is that taking data from different sources and different modalities, we're able to integrate it and better understand patients. And so this, as an additional step to a screening process, can really provide huge benefits for our collaborators, especially within pharma and clinicians. And also, please feel free to raise your hand if you'd like to say something live. Just out of curiosity on my part, raise your hand if you are a clinician. I'm interested to see who has joined us today. I see a couple. Great. And so with the clinicians present, I'm interested to know, and I'll unmute you and you can feel free to talk. If what are some of the problems that are most pressing to you that you think comes from a lack of understanding your patients in a very personalized way? Feel free to answer, feel free to not, and I could definitely have this discussion with Christian as well.
I see, Doug, that you're one of the attendees. I think it would be interesting to have your take or your point of view on this.
Not to put you on the spot.
Yeah. So, I mean, from my perspective, it's you want to know a patient's prognosis so that you can individualize the approach to their treatment. You also want to be able to predict if they're going to respond to a particular intervention.
That's a great point, Doug. Christian, do you have more to say about that?
I mean, I totally agree. This is essentially why those two strategy of prognosis and predictive enrichments are framed for. Like, it's really to try to pinpoint those two aspects that Doug highlighted. So I don't have anything to add on that. I think it's a difficult task that definitely needs careful attention. We will definitely not be able to achieve like perfection on these tasks because there is a lot of information and components that are or information that is not available to us or to the model when he's doing his prediction. But I think if we can be better than chance, there is definitely a lot of value to take into account that types of prediction, either on the clinical side or on the clinical trial side.
Great. So here's another question. Building on what Dr. Bengio said earlier this morning, what differentiates successful and unsuccessful applications of AI in the clinical space?
Yeah, it's a good point. I think validation is a critical thing. We need to validate and replicate across multiple data sets these types of tools to make sure that they do generalize once they are deployed either in the clinic or in clinical trials. That also link to the different types of settings where you acquire the data. So if it's imaging, you may have like multiple types of scanner and variability or heterogeneity that comes with it. And you need to have ways to model that heterogeneity or at least be robust to that type of heterogeneity that is not the patient heterogeneity, but really more acquisition scanner or the type of modalities that you're acquiring. So I think these two aspects needs to be carefully validated and replicated to be able to have successful AI algorithms deployed. And this is even more true in the clinic where you have a much more broad population of individuals that will have that may not necessarily correspond specifically only to the scope of the clinical trial.
Great, thank you. So here's a question from Joseph Viviano. How will this platform work with trial pre-registration? Will the predictions of the model be used when submitting the trial applications or after the trial is approved? If so, will the regulatory bodies be able to audit the potential bias in the predictions of the model when deciding to approve the trial?
So we intervene, so it's a good question. So we don't, the way we envision this is to be during the screening period of the trial, so before randomization. So it's a really clean process during the clinical trial. We don't intervene at all. The population that is selected by the inclusion-exclusion criteria that are normally done during the trial, plus you can see us or a prognostic enrichment criteria being another inclusion criteria or exclusion criteria. So the normal framework of a clinical trial will still be respected, and we don't have any peek into the after randomization. So basically, this is ongoing discussion that we have with the FDA on how they want to regulate that. But basically, whatever criteria that you have, inclusion, exclusion criteria to characterize your population that is investigated should be tied to the indication of the drug. And therefore, like if it's an algorithm or a threshold on some variables, then those things will be the characteristics of the population that have been investigated. And if the drug is approved, that's the definition of the population you should treat.
Thank you so much. So here's a question from Nathaniel. How large are the populations we screen, and how rare are the events that we would detect? He asked because in typical biomarker-based screening efforts, rare events in large populations tend to lead to overtreatment. Many more false positives receive unnecessary treatment than there are true positives receiving benefits.
So depending on the disease, the prevalence will differ. Individuals and depending on the stage of the disease also. So in the more severe phases of Alzheimer's, you may have greater than 30% of individuals that will not decline during the trial. Less than 30% that will not decline. And in mild cognitive impairment, so individuals that starts to have symptoms but are not demented yet, you will have around 30% or even more of the individuals that will not decline. In cardiovascular, it's going to be around 10% of individuals that will actually have the event that you decide to reduce in secondary event, and before that, before an initial event, then those proportions increase. So it really depends on the disease. Obviously, like that's why also we are not taking extremely rare events. Like we need examples and a case where there is actually a substantial amount of individuals that will have the desirable events.
Cool, thank you. So Sandeep has a question. Have you considered medical records matching AI company pre-fill your list, and then our platform could produce a highly qualified patient slash participant list? This could potentially almost eliminate a recruitment advisor.
Yeah, so that's the intent of where we want to intervene, basically. Like you have a first wave of screening with the inclusion-exclusion criteria, ideally in an automated way. Currently, there is a lot of manual work related to identifying those individuals based on the inclusion-exclusion criteria, and then we follow with our analysis once you acquire the relevant biomarkers for those individuals that fit the inclusion-exclusion, the minimal inclusion-exclusion criteria. So yeah, it would potentially be much more automated, and you can source larger database and potentially come up with candidates that are more suitable and also faster.
Great. So here's another one. Outside of clinical research, where do you see these approaches being applied to provide meaningful benefits within patient journeys?
Well, I think from our experience and our research in the clinic, it seems that most of the drugs don't have a perfect efficacy. And therefore, I think there is room to use tools like that to pinpoint a population of responders and therefore expose patient to the right treatment and prevent others of being exposed to potential risk if they don't have a high likelihood of benefiting from that treatment. So that's one thing. I think maybe Trevor, you have some ideas more on the payers and providers as well.
Sure. So we're in Canada, so there is one payer and it is the government. But in the US, there's a variety of different payer models, many public, many private. It can include Veterans Affairs Healthcare. It can include Medicare, which is different in the US. It generally means people under a certain income level. And also private insurer networks that have their own hospitals and also employer-based insurance. So there's a ton of different models. And so better understanding the relative risk of different patients and as branching out from that, predicting costs can have a huge amount of benefits in both patient outcomes and also to the payers themselves. There's a new and increasingly used paradigm of value-based care, which is reimbursements in the healthcare field being based on the quality of care rather than the amount of patients, which generally produces better outcomes, but also requires a really good understanding of relative patient risks and their outcomes. And so we definitely see a bunch of potential applications in that field as well. And also eventually in the clinic, as Christian said.
I think there is a lot of room to grow in the clinic in various applications. There is already a few examples of that, but this is very nascent field. I think we will see a lot of emergence of newcomers and ways to optimize response to treatment of drugs that are currently on the market using real world evidence. I think that's going to be a very interesting wave of new solution that will come to the market in the coming years.
Excellent. And this is my question, but just building on that, where do you see this technology leading in the next 2 years, the next 5 years, and if you have an answer, the next 10 years?
So as I said, this is like a very new field, but I think that has like clear value and like clear insight that you can derive from such a prognostic information. I really think that the starting point is clinical trial, where like it's a more rounded and clear framework, where we can carefully validate those tools and then bring them to the market or to the clinic once they are approved, either as companion diagnostic or as a tool that you can use alongside the drug or as an independent tool. So I think that's the first wave is in the clinical trial setup, and then much more application can be derived in the clinic with real world evidence actually that will increase another. That's also maybe a point that I didn't cover, but most of these tools have characterize a certain population, and we can potentially refine those tools once the drug is approved and in phase 4 or evaluated in the clinic. It's not like we don't do any study anymore. We're still trying to monitor the drug efficacy and how it's administered and to which population. And I think this should be a more dynamic circle. Basically, we should iterate on that and see what's the impact of the population being treated, and try to refine which population we want to optimize for, and identify the subjects either that will not respond or will not have or the ones that will respond.
Great. So I think we have time for one last question if anyone wants to reach out.
You can raise your hands.
Yeah, feel free to raise your hand. All right. Well, I will ask one last question. So from your perspective, what are some of the biggest obstacles that the pharma industry is facing today? And how can our platform really alleviate that? So I guess that's really, in a nutshell, how are we helping pharma remove these obstacles to getting therapies to patients faster?
I think Yeshua alluded on that earlier. Basically, data is key in these types of analysis, and therefore we need to share the data and try to find ways to cut part of the red tape while preserving the identity of the individuals that do contribute to that data. So we need to find ways to better manage that and provide that information so that we can actually train competitive models that will have that will be as less biased as possible, and really deliver on accurate predictions.
Great, thank you so much. So if anyone has any questions stemming from this discussion, please don't hesitate to reach out to either myself or Christian, and we're more than happy to connect with you on LinkedIn or offline. So thank you so much for attending. This has been a great discussion. Thank you so much for your excellent presentation, Christian, and we'll see you at the conference.
Thank you very much, everyone.
Take care.